Welcome to Brain Ponderings. I'm Mark Matson. This is the second in a series of presentations on brain bioenergetics and health. And this one's going to focus on mitochondrial dynamics, how mitochondria move to different areas in neurons, how they respond to, for example, activation of synapses, the mechanisms that control the division of mitochondria. Are there fusion of two different mitochondria, as well as a process called mitochondrial biogenesis, the generation of new mitochondria. I'll also talk finally about how cells, particularly neurons, recognize and eliminate dysfunctional mitochondria to make sure that all the mitochondria in the cell are healthy and producing ATP and carrying out other functions. I'll also mention a few things about how mitochondria play roles in signaling within a cell and maybe even between cells. And then next time, I'm going to focus on how exercise and fasting affect the brain, an area that my lab worked on for more than 30 years, which is very interesting. And I'll kind of touch on a lot of the things I've already talked about in these first two presentations in terms of how exercise and fasting affect those on the one hand, and how this all relates to the beneficial effects of exercise and fasting on brain health. So I'll go ahead and share my slides, and just a reminder for these presentations, I am showing slides. So if you want to see those on my YouTube Brain Ponderings channel, you can do that. Okay, so this first image here is a three-dimensional reconstruction generated from serial electron microscope images, serial sections, in of a dendrite. So that's shown here. This long structure here is the dendrites with dendritic spines projecting out various places. And then shown in light yellow here are presynaptic terminals, right? So this is where the neurotransmitter is released and acts on the postsynaptic receptors on the postsynaptic dendrites. So in this particular image, there are 1, 2, 3, 4, 5 presynaptic terminals. And they're associated with spines in general. Here's one dendritic spine that there's no obvious presynaptic terminal, which can actually happen in some cases. There are dendritic spines where there aren't synaptic terminals, but it's rare. Okay, shown in color here in pink, and of course, everything is on a grayscale when you look at these images, raw images from the electron microscope. So in this case, the mitochondria, individual mitochondria were colorized, and the dendritic mitochondria, shown here in pink, this one's very long, extending through a large region of the dendrite. And then the presynaptic mitochondria, shown in green, tend to be much smaller. But anyway, the point here is that mitochondria are localized where the action occurs in terms of neurochemical signaling within neural networks, which makes sense because they have to be able to provide the ATP necessary to drive these energetic processes. Whenever your nerve cells are active and firing, there's a big increase in energy demand. Okay, so in this next slide, in the upper right corner here is a single neuron, embryonic neuron from the hippocampus of rat embryos, and it has been in culture growing on a flat substrate for 3 days approximately. So when this neuron is initially placed in culture, it's a sphere. So during the 3 days, the dendrites have grown and the axon has been established and grown faster than the dendrites. And this image, actually, I took this image when I was a postdoc at Colorado State University in the 1980s. And I was doing experiments and showing that glutamate, the major excitatory transmitter in the brain, controls the growth of these dendritic growth cones and controls synapse formation during brain development. But anyway, this kind of nostalgia here, I used a 35 millimeter camera. In this case, it had color film. There was no computer then that we could hook to a microscope and acquire digital images. So a lot of the work I did had to do with actually taking pictures of these neurons at different time points and then quantifying the growth after I developed the film and then projected the images. But anyway, so on the left here is a schematic diagram showing mitochondria. Um, and so, of course, there's mitochondria at all stages of growth during development. But as the neurons grow and extend their processes and differentiate in axon and dendrites, the number of mitochondria increase and they're moved and distributed throughout the axon and dendrites. And then when synapses form, they're present near synapses. In the lower left here, these are the red is mitochondria. So in these experiments, the neurons were living neurons were stained with a fluorescent molecular probe that is selectively taken up into mitochondria based on the hydrogen ion gradient, the proton gradient across the intermitochondrial membrane. But the main point here is to show that this is a neuron 3 days in culture, and this is another neuron after 10 days in culture. And you see by 10 days in culture, where you've got elaborate dendritic arbors grown out, you have mitochondria distributed throughout them. So the number of mitochondria increase. That's a process called mitochondrial biogenesis. Okay. So first I'm going to talk a little bit about how mitochondria are moved to different parts of cells. And right. So the key thing here is they are moved along kind of railroad tracks, if you will, of molecular polymers called cytoskeletal proteins, and in particular, uh, microtubules. So there's two main uh, components of the cytoskeleton. One is microtubules, which are made of a protein called tubulin that are connected in polymer. So a chain, in this case, the chains of the tubulin polymers wind and form a tube. So these are thicker than the actin filaments, the microfilaments, which are made of the protein actin. And both microtubules and microfilaments are dynamic in that they can grow in length by adding either tubulin polymers to the microtubule end or single actin molecules to one end of the actin. Okay, and actually the three-dimensional structure of these proteins, the actin, and this is the actin monomer, and this is tubulin dimer, two tubulin molecules has been worked out. Kind of interesting. So I put that in. It's just to show you there's been a lot of work done on these cytoskeleton proteins because they're so important in cell function and moving things around cells, and actually critical for the growth of cells. For example, the growth of an axon or dendrites requires uh, polymerization of these. Okay, so mitochondria can be moved along microtubules, and that's shown here in this example where there's an enlargement of an axon of a cell here showing 3 mitochondria, and they can be moved either away from the cell body towards the end of the axon That's called anterograde transport, where they can be moved back towards the cell body. It's called retrograde transport. And there are energy-dependent motor proteins that link the mitochondria to the microtubules and move them. So for anterograde transport, there's a protein called kinesin, which is the motor protein for moving a mitochondria this way. And for moving mitochondria in the retrograde direction, dynein. So kinesin and dynein are the motor proteins. Um, and there's also a role for actin, the microfilaments, in controlling the movement of mitochondria. And essentially, the actin filaments, they can stall or stop the movement of mitochondria. And turns out the actin filaments are abundant, particularly uh, in the synaptic regions. For example, here's a dendritic spine with actin filaments shown in purple here. And so what can happen is when this synapse is activated, say, for example, glutamate is released from a presynaptic neuron, activates the receptors in the membrane, resulting in calcium influx. Uh, then that calcium promotes the polymerization of actin, and then that stalls the mitochondria, makes them stop at that synapse, which kind of makes sense because when the synapse is very active, you want a lot of energy, ATP present, to restore the ion gradients, for example. And so this is kind of an interesting mechanism where the cytoskeleton influences where the mitochondria are in response to the demands, the activity of that particular synapse. Yeah, that's kind of the key things there. So this next example shows up a little closer. So I mentioned kinesin and dynein, the motor proteins. So they interact directly with the microtubules, the tubulin, but they don't interact directly with the mitochondrial outer membrane. There are proteins, so-called adapter proteins or linking proteins that kind of bridge, connect these motor proteins to the mitochondria. In the case of kinesin, it interacts with a protein called miro. In the case of dynein, it's thought to interact with a protein called VDAC in the mitochondrial membrane. But the point here is that this is the way these interactions occur, the kind of physical interactions that attach. And then these work kind of like in muscle cells where you get this ratcheting of the actin and myosin in the muscle cells. So these things kind of, go like this. They move and then release, move, release, move, release. So that can move the mitochondria along. And the mitochondria move uh, on time scales of like maybe 1 millimeter in a minute or something like that. 1 to 10 minutes. I mean, they can move at different rates, but they're kind of in that ballpark. So it's not like they're zipping from the cell body to the axon terminal real quickly. Okay, this just gets into a little of protein structure. So it turns out this is a miro, this protein that attaches the kinesin to the mitochondrial membrane. And it has these structures called EF hands, which bind calcium. So just like in muscle cells, where the movement of the actin and myosin is stimulated by the calcium influx when the muscle stimulated and contracts, this movement along the microtubules can be affected by calcium. And that's shown on the right here. Okay, also, if one looks at this in a little more detail. There's some differences in the transport in axons and dendrites. In axons, the mitochondria, these motor proteins, move them only in one direction, whereas in dendrites, the mitochondria can move in both directions more easily with one or the other motor protein. Okay, so I mentioned that when you get calcium influx of the synapse, the actin can polymerize and interact with the mitochondria and stall them so they stay at the synapse when it's active. And there's a protein called syntophyllin that mediates the interaction of the uh, uh, that responds to the calcium and is also involved in stalling. All right, interestingly, and this is this image I showed you at the beginning. Um, there's been studies, including by Pamela Yao and Tom Delgado in my laboratory before I retired from the NIH. And this is their images they generated from the electron micrograph. So on the upper is a single thin section showing, again, presynaptic terminals in yellow, and in this case, postsynaptic regions in blue. Uh, and they did they looked at essentially they looked at the size and morphology of mitochondria in axons versus dendrites and compared them. And what they found was that in every brain region they looked at, the mitochondria in the presynaptic terminals, the axon terminals, were relatively small and stubby, whereas the mitochondria in the dendrites were long. Uh, and so that's pretty interesting. And we think actually this has something to do, interestingly, with communication between synapses in one dendrite with the mitochondria. So you see in the dendrite, you've got one mitochondria and many synapses impinge indirectly on that mitochondria. So we think that there's some role for mitochondria in coordinating activity in synapses that are adjacent to each other. And in a little bit, I'm going to talk about how this might work with regard to calcium, because it turns out that mitochondria can take up calcium from the cytosol, they can release calcium, and that this uptake and release is regulated and may actually play a role in what we call synaptic plasticity, which seems to be the underlying basis of learning and memory. All right, so anyway, these difference between axon and dendrite mitochondrial morphologies occurs in all brain regions. This is the nucleus accumbens, mitochondria from neurons there. This brain region's been studied a lot for its role in addiction because it's neurons there that the dopaminergic neurons that release dopamine act on. Uh, this is examples of mitochondria in a region of the hippocampus called CA1 that's critical for short-term memory, and neurons in this brain region die in Alzheimer's disease. Selectively, they're ravaged, very vulnerable in Alzheimer's. Um, Here's examples from the cerebral cortex where there's even a huge difference in size between the axonal and dendritic mitochondria. And this is a nucleus involved in the auditory system, the hearing system, sound. So these are the neurons mitochondria in the neurons that send the impulses from your ear to your auditory cortex. All right, so this kind of a summary of all this. We've got long and tubular mitochondria in the dendrites, um, and there's roles for calcium, which I'll talk about. And I mentioned energy, field local translation. So there's evidence that, well, By translation, they mean the production of proteins from messenger RNAs. So remember, genes encode for messenger RNAs, which are then the sequence of the mRNA is translated into amino acid sequence. So that's called protein translation, and that's a energetic process that occurs on ribosomes. And there is evidence that when a synapse is active, it can stimulate the local production of certain proteins. For example, one that I'll talk about a lot is called brain-derived neurotrophic factor. So its levels it can be produced locally uh, in dendrites, and there's a role probably for mitochondria in influencing that. And that BDNF in turn is then released and promotes the increase in size of a synapse, formation of new synapses, and it plays a role in learning and memory. All right, so if that's not complicated enough, mitochondria have even more complicated roles besides producing ATP and regulating calcium. And there's some really interesting work that's been done in looking at mitochondrial membrane dynamics. So remember, mitochondria have two membranes, an outer membrane, and then the inner membrane where the electron transport chain complexes are, where the ATP is produced. And that inner membrane is invaginated and has a big surface area compared to the outer membrane. Okay, so it turns out that mitochondria interact very closely with other organelles in cells. And one organelle that's been studied extensively in terms of its interaction with mitochondria is called the endoplasmic reticulum, or ER. Now, there's two types of endoplasmic reticulum. So these are membranous structures, as shown here in pink, and they're often kind of long and thin and have different compartments within them. So the two types of ER are called rough endoplasmic reticulum, and that's where a lot of protein synthesis uh, occurs. And then there's ribosomes associated with that uh, so-called rough ER, and then the proteins are produced in the cell and then ultimately get out and can be moved to any region of the cell. Now, the membrane of the ER is often closely opposed, adjacent to the outer membrane of the mitochondria. And this ER, particularly the so-called smooth ER, is known to be an important mediator of calcium signaling. So when, for example, a synapse is active, calcium is taken up into the endoplasmic reticulum. It can be stored there, so there's higher concentration inside the ER compared to in the cytosol of the cell, gray area here. And then that calcium can be released in response to certain signals. For example, the neurotransmitter acetylcholine can be a potent stimulator of calcium release from the endoplasmic reticulum. And then that calcium can be taken up by mitochondria. And then they can, in turn, when they get certain signals, release the calcium. And this whole calcium regulating system is sensitive to energy levels. So, for example, if you just take cultured neurons and you subject them to a very low glucose level, so you deprive them of energy, that will affect this interaction between mitochondria in ER and calcium levels in the cells. And in turn, that change in calcium leads to the activation of genes that encode proteins that help the cell tolerate the low energy conditions. Okay, in a little bit, I'll talk about how mitochondria can be when they become dysfunctional and aren't generating ATP well, maybe they're producing excessive amounts of free radicals and have a lot of damage to the components of the mitochondria. A dysfunctional mitochondria, or even a dysfunctional part of a mitochondria, can be recognized by certain molecular machinery and moved to the lysosome, the acid bath, and then removed. So there's this we call that mitophagy. So it's a kind of subtype of autophagy. If you didn't see my conversations with David Rubenstein at Cambridge University, who's an expert on autophagy, you might want to see those two podcasts where he does a really good job about first explaining autophagy and mitophagy, and then in the second episode talking about how impaired ability of cells to engage this autophagy machinery that removes molecular garbage and dysfunctional organelles, how that impairment contributes to Alzheimer's disease, Parkinson's disease, and actually occurs somewhat during normal brain aging. Okay, so now I'm going to talk about mitochondrial fission, that is uh, splitting of mitochondria in two, and mitochondrial fusion, the merging of two individual mitochondria. I'm going to talk about that just a little bit quickly. Uh, the molecular machineries for those have been worked out in large part, and that those molecular machineries have been manipulated using genetic tools to change the levels of the proteins involved in fission or fusion, and then look at the consequences for cells. Um, right, and I mentioned this in my last presentation that in all of these slides, uh, unless I missed it, hopefully I didn't, I cite the journal article from which I took these illustrations. In this case, it's an article in Nature Reviews Molecular Cell Biology 2020, Volume 21, those pages. And most of these can be accessed free on PubMed. So if you just go in and put this in there and hit return on your keyboard, you should get that article free. So you can read the text going where they talk about this in detail. So for you scientists out there, neuroscientists who aren't so familiar with these things, if you want a lot more detail, you can go to the articles. All right, so fission, the division of mitochondria. Okay, so there's a protein called DRP1, dynamin-related protein 1, that is critical for the fission, the splitting of one mitochondria into two. And this process involves the actin cytoskeleton. So this is kind of a low magnification image here showing the endoplasmic reticulum kind of wrapping around the mitochondria here and the DRP1. So here we have high magnification of this showing the endoplasmic reticulum tubule here, if you will. So this is a membrane here, and then there's actin filaments. There's some intervening proteins between the actin and the membrane of the ER, and another kind of adapter protein, linking protein between the actin and the membrane of the mitochondrion. And then this DRP1, together with this actin cytoskeleton, essentially kind of physically constricts the mitochondria in the middle here. And eventually, the membranes of the mitochondria, outer membrane, splits in two, essentially, and then you have two mitochondria. Okay, in the case of fusion, and one thing that if you get high level of calcium influx, that can cause a splitting of mitochondria. Then fusion, there's a protein called mitofusin 1. Okay, that's a logical name. And it's critical for the merging of two mitochondria. And higher mag here shows some of the intervening proteins. And this is a energy dependent process. All these processes require the energy in phosphate bonds from ATP, for example, or GTP. All right, now I'm not going to spend much time on talking about this slide here. It has to do with the fact that these various proteins that are involved in mitochondrial fission, fusion, uh, even the transport and movement of mitochondria along microtubules are interacting with the actin cytoskeleton. Those various proteins, their functionality can be affected by various signaling pathways by what is called post-translational modifications. So a very well-known post-translational modification that mediates responses of cells to various stimuli, whether it's a neurotransmitter or a hormone or even electrical activity. So pretty much any environmental challenge. One of the main post-translational modifications is called phosphorylation, where the phosphate in ATP, one phosphate uh, atom its phosphate molecule is transferred to uh, particular amino acids on the protein. In fact, there's three amino acids only that seem to be phosphorylated. One is serine, one is threonine, and another is tyrosine. And there are enzymes that transfer the phosphate from the ATP to the protein. Those are called kinases. And there are also enzymes that can remove the phosphate from the protein. Those are called phosphatases. Okay, so these kinases and phosphatases can be activated by various signaling pathways, by neurotransmitter, glutamate, dopamine, serotonin, hormones, and and uh, and and intracellularly, calcium is one important regulator of these kinases and phosphatases, but there are others. In fact, some receptors themselves, the receptor itself has kinase activity on the inside of the cell membrane. Okay, so phosphorylation and these various proteins involved in fission and fusion uh, can be modified by phosphorylation. Another post-translational modification is called ubiquitination, where a small protein called ubiquitin is added to the protein, and this is actually very important in autophagy or in moving uh, damaged proteins to be degraded uh, by various means. Okay, and I'll also talk about later on, I'll mention at the end of my presentation today, there's a process called acetylation, where an acetyl group uh, can be added to a protein or removed by um, acetyltransferases or acetylases. And this turns out to be very important with regard to brain bioenergetics because things like exercise, fasting can affect this process of acetylation in ways that bolster the resilience of neurons and enhance their functionality, and in fact, over time may keep them functioning well uh, throughout the lifespan and protect against disease. Now, there are certain genetic disorders that are caused by mutations in proteins involved in mitochondrial dynamics. So shown here is examples of mutations in DRP1. So mutations that impair fission of mitochondria. Uh, Also, mitofusin 2. So there's certain disorders that involve impairments in fusion of mitochondria. And these all have neurological phenotypes. So they affect the nervous system. For example, DRP1, certain mutations have dramatic effect on brain development. So the individual is born with a lot of problems with the brain. There are others that seem to affect certain subcomponents of the nervous system. For example, the optic atrophy syndrome affects vision. This one, pain, sensitivity, and so on. So again, we see that mitochondria seems to be kind of perhaps they're important for every cell, but they're particularly important for neurons, which have these elaborate morphologies. They have to be moved around a lot. They have to increase the amount of mitochondria and respond to demands. And so any problem with mitochondria, or a lot of problems with mitochondria, manifest with neurological symptoms. Uh, yeah. So now I'm going to talk about the removal of mitochondria, how cells recognize that a mitochondrion isn't working well, and remove it by sending it to the lysosomes. And you can imagine you have a long axon, could be a foot long or more. And then remember, this for scale here, a cell body of a neuron is typically 1/100th of a millimeter in diameter. The uh, diameter of an axon actually is typically 1/1000th of a millimeter. Uh, and then, so you have a neuron that can have an axon of a foot or more. And you can imagine that you have mitochondria distributed all along that, and there's some on the end here. And it's thought that most of the mitochondria that become dysfunctional have to be transported back to the cell body to be degraded in the lysosomes. Um, Although there's kind of emerging evidence that there may be lysosomes out in the processes, but they're not very obvious when you look at this, for example, by electron microscopy. Okay, one interesting thing that's been fascinating to me for a long time is that has to do with both stem cells and it has to do with mitochondria, and that is that individual stem cells or individual mitochondria have mechanisms whereby, and remember, stem cells are essentially immortal, um, or can be immortal. And so what these cells and what mitochondria apparently can do is that they can recognize and move damaged and dysfunctional components to one end of the cell or mitochondria. So it could be damaged proteins, it could be dysfunctional mitochondria or damaged endoplasmic reticulum, damaged membranes. For example, oxidative stress can cause damage to membranes. And this was discovered initially in yeast, the budding yeast, which these cells divide what's called asymmetrically, and that's where this segregation of damaged and undamaged components occurs. And what was shown in these yeast is the damaged components are moved to one end of the yeast cell, and cytoskeleton's involved in that. And then that part buds off the yeast. So you're left with a mother cell, stem cell, if you will, that's very healthy, happy, and has very little damage or no damage. And you have a daughter cell that has a lot of damage. And then these cells, daughter cells, they live a while often, but they eventually die, whereas the mother cell keeps going on and giving rise to these daughter cells. And this has been worked out how this works at the molecular level, the different proteins involved. This seems to be also true uh, in mitochondria, where you can have segregation of damaged components, then fission, and then the damaged daughter cell, if you will, daughter mitochondria, is recognized as being damaged and moved to the lysosomes and degraded. So that's pretty cool. I also want to point out, so I mentioned next time I'm going to talk about exercise, fasting in the brain. And then in the final presentation in this series on brain bioenergetics, I'm going to talk about bioenergetics in relation to brain aging and neurodegenerative disorders, which is something we also worked a lot on in my lab over the years. Um, and I just want to mention this here, and I'll talk about it when I get to the neurodegenerative disorders, but Parkinson's disease has proved to be a disorder that is focused heavily on problems in the mitochondria and the problems in mitophagy, in recognizing and removing dysfunctional mitochondria. And the reason for that is that there are rare cases of Parkinson's disease that are caused by mutations, gene mutations, and usually individuals that have those mutations develop symptoms at an early age, even in their 30s, 40s, and of course they eventually die from Parkinson's disease. And these different genes, so Parkin is one, Pink1's another, there's another one called DJ1, another one called LRK, and these have all been shown to play some role in either mitochondrial function or mitophagy. And also, there's kind of environmental toxin-based reason to believe mitochondria play a role, and that is that there's certain mitochondrial toxins, like the insecticide rotenone or this chemical MPTP that was discovered serendipitously, that they act directly on mitochondria at the molecular level to impair electron transport and cause excessive free radical production. And when you give those uh, neurotoxins to mice, rats, monkeys, or in case of accidental poisoning, humans, they develop symptoms that a neurologist, it's indistinguishable from Parkinson's disease, and they have selective death of the dopaminergic neurons, which die in Parkinson's. Okay, I just wanted to point this out. I'll go into it in more detail later, that these processes, there's evidence they can be affected by um, exercise and fasting, and also by using your nerve cells, exercising your nerve cells, not just physically exercising. And one way that occurs is through the process of mitochondrial biogenesis, where you have a healthy mitochondria that has very little or no damage, and it divides, and then the two mitochondria arising from that single mitochondria grow in size, and can, so you get an increased generation of ATP, and yeah. Right. Okay. I'm not going to go into this in detail. This is a diagram actually taken from a chemical company's website. Um, and they often, actually, it's interesting. They have some nice charts of different metabolic pathways because they often sell chemicals that influence some particular component of these. Um, but anyway, I just want to point out that this process of mitophagy uh, is influenced, autophagy is influenced heavily by bioenergetics. So fasting will stimulate mitophagy and autophagy. So that enhances the removal of molecular and organelle or garbage, dysfunctional mitochondria. And these pathways have been well described. So I just wanted to show you that here. Okay, mitochondrial biogenesis. So this was first discovered, the mechanisms that control it were discovered in muscle cells and how they respond to exercise. Uh, and what was shown was that with exercise, your muscles are contracting. They're contracting because they're being stimulated by the nerve cells that innervate the muscle cells. And there's calcium influx and also actually calcium release from the endoplasmic reticulum. And that calcium in muscle cells and nerve cells will activate certain kinase, which I mentioned transfers a phosphate group to target proteins. That's actually shown here. So this kinase is called calcium calmodulin dependent protein kinase 4, but the calcium binds to a protein called calmodulin, which then activates this kinase, which transfers the phosphate group to a number of different proteins, one of which is called CREB, which is a transcription factor in the muscle cells and we think in nerve cells it plays an important role in mitochondrial biogenesis in response to activity of the cells. So how does CREB do that? Well, it in turn stimulates the gene encoding protein called PGC1 alpha. And this is a transcriptional regulator that controls the production of a whole series of other proteins that have to do with importing newly uh, translated proteins into the mitochondria. Remember, most of the proteins in the mitochondria are encoded by nuclear genes. So when you exercise, you're getting increased production of new mitochondrial proteins that are then moved and imported into the mitochondria. So that makes sense. In order for mitochondria to grow and keep growing, divide, you have to have more proteins. Um, and in addition to calcium influx, this pathway, PGC1 alpha, can be increased in response to reductions in ATP levels in the cell. So there's a kinase uh, called AMP kinase. It's activated by adenosine monophosphate. So uh, when ATP is adenosine triphosphate, right? Three phosphate group. So when ATP is being used up, you get an increase in AMP, and that triggers activation of PGC1 alpha. Okay, but but the process of mitochondrial biogenesis is actually not occurring while you're exercising. It's occurring during the recovery phase. But these events occurring during exercise set in motion uh, the events that enable the growth and division of mitochondria through this pathway. And I'll talk about this a lot next time, that these cycles of metabolic challenge, recovery. So exercise, resting, eating, sleeping. Resting, eating, sleeping is the recovery. Fasting, that's the metabolic challenge. Recovery, again, eating, resting, sleeping. These cycles of challenge recovery over time promote the growth and resilience of cells. So in the case of muscle cells, depending whether you do aerobic or resistance training, increase endurance, increase strength, or both. In the case of nerve cells, there's an increased number of synapses, uh, increased growth of dendrites of neurons, for example. There's evidence for that with exercise fasting. Even increased production of new neurons from stem cells, process called neurogenesis, which I've had several podcasts on. Okay, and this is just another way of looking at this. Again, uh, oh yeah, this is important too. This can also be affected by insulin signaling, okay, in interesting ways. And that gets to be important when you talk about when we talk about uh, fasting, where you get a switch from using glucose to ketones. All right, so I'll just show you some data from a study done by Wu Cheng, who was a senior member of my lab for 20 years at National Institute on Aging. And she's very good with a lot of different technologies. Uh, she's done a lot of work both in cultured neurons and in mice, in particular living mice, genetically modified mice. So I'll show you some data relevant to mitochondrial biogenesis. Okay, so this slide just shows that I already mentioned that as these neurons grow um, during brain development, the number of mitochondria increase, and the amount of ATP as expressed as relevant to total protein, total cell protein increases. So you're getting increased number of mitochondria per cell and increased ability amount of ATP they can generate. All right, so that what Dr. Cheng did, she used a genetic tool called RNA interference. What this is it was discovered that actually cells produce what are called small interfering RNAs that can affect the translation of proteins by binding to the messenger RNA. So what Dr. Cheng did, she used a adenovirus, which is a non-toxic virus that infects neurons, and she put in the DNA of the adenovirus uh, sequence, nucleic acid sequence, complementary to the messenger RNA of PGC1 alpha. So what's happening here is when the neurons are infected with the virus produces this called small interfering RNA or siRNA, and then it prevents the production of the PGC1 alpha protein. So the bottom line is this is a tool where we can control the level of PGC1 alpha in these neurons, and presumably, and as I'll show you, it's true, we can reduce the amount of PGC1 alpha and reduce mitochondrial biogenesis and see what consequences that has for the cells. So she also had uh, in that virus, she had the genes encoding a green fluorescent protein. So that it turns out not all neurons are infected when you give a certain amount of virus. And so, which is kind of nice because then some of the cells are infected and have reduced PGC1 alpha, whereas other neurons in the same culture aren't infected and have normal levels. So you have the experimental and control in the same culture. And what she did is then she quantified dendritic spines, which is a indirect measure of synapse numbers, and she found uh, indeed when she reduced PGC1 alpha, it decreased the number of dendritic spines. It also decreased um, number of mitochondria because there's impaired mitochondrial biogenesis, and it also reduced uh, ATP levels. Okay, so then, so that suggested that mitochondrial biogenesis is necessary for the formation of new synapses. during brain development. And she looked at this in other ways, actually looking at uh, presynaptic levels of a presynaptic protein, postsynaptic protein, PSD95, and a presynaptic protein, synapsin, which is involved in neurotransmitter vesicle movement and neurotransmitter release. And so there's decrease in both post and presynaptic markers. And she did that by quantifying from immunostain where she had antibodies against PSD95 and synapse. And she also did immunoblots and found that when she could overexpress, she had a virus to overexpress PGC1 alpha, she could increase number of synapses. So you can artificially accelerate synapse formation by increasing mitochondrial biogenesis. Okay, so that's in developing neurons, embryonic rat neurons. What are mouse neurons, actually, in that case? What about in an adult mouse? So she injected the virus to reduce PGC1 alpha levels in glutamatergic in neurons in the dentate gyrus of the hippocampus, most of which are glutamatergic neurons. And then after these neurons were infected with the virus to reduce PGC1 alpha levels, uh, she let the mice go live for another several weeks. And then she euthanized the mice and looked at the synapses, the dendritic spines, and found that in living animals, over a period of several weeks, the number of synapses is reduced when the neurons are not able to generate new mitochondrial biogenesis. So that shows that mitochondrial biogenesis is necessary for the maintenance of synapses in the adult brain. Okay, then to look at this in more detail, it had been known, other labs had shown a long time ago that BDNF, this neurotrophic factor that's stimulated potently by physical exercise, it's also stimulated potently by activity in neurons. So hopefully, for sure, in my neurons in my brain that are enabling me to think about what I'm talking about, remember what I know, tell you what I know, uh, and in your brains when you're trying to understand this, it can be, you have to think a lot. So nerve cells are more active in the circuits that are involved in that thinking. BDNF levels increase. That's good because if you keep your mind intellectually engaged on a daily basis over time, probably you're going to increase number of synapses or maintain them for longer as you get older. And there's good evidence actually that people who keep intellectually challenged uh, during their life are less likely to develop Alzheimer's disease, for example. Anyway, so we asked the question, there was evidence that exercise increases mitochondrial biogenesis in muscle cells. We've shown that mitochondrial biogenesis is important for synapse formation. It had previously been shown that BDNF is critical for synapse formation during development and maintenance of synapses in the adult brain. And the obvious question was, is mitochondrial biogenesis necessary for the ability of BDNF to promote the formation and maintenance of synapses? And so I'm not going to go into the details on this slide. If you want to look at it, it's in this Nature Communications paper in 12 years ago that we published. So the first thing she asked was, will BDNF stimulate PGC1 alpha uh, increase in the neurons? And the answer is yes. Okay. And then and that's shown here looking at the various markers of mitochondrial biogenesis. TFAM, I think I mentioned before, is a transcription factor involved in transcription of genes encoding mitochondrial proteins. And she also showed that BDNF increases number of mitochondria. Okay, then the key thing was if we reduce PGC1 alpha levels using the RNA interference technology, will that affect the ability of BDNF to to stimulate synapse formation? And the answer is yes. Okay, right. Now, another lab, actually quite a while ago, had provided evidence that in cultured neurons, BDNF can stimulate energy metabolism and increase glucose uptake into the cells. So BDNF can promote glucose transport into the cells. Okay, I'll finish up now talking about signaling, mitochondrial signaling functions. And I mentioned this interaction with the endoplasmic reticulum, and that calcium can be taken up and released by mitochondria, and that can be affected by activity in neurons. Now, remember, the excitatory neurotransmitter in the brain is glutamate. More than 90% of the neurons in the brain deploy glutamate as their neurotransmitter, and it is a potent stimulator of calcium influx at the postsynaptic sites. Now, a long time ago, actually in 1986, the late Roger Chen, Nobel laureate, he developed a fluorescent probe that can be introduced into living cells, and that its fluorescence changes when in relation to the calcium concentration in the cells. So if you and it turns out that my mentor when I was a postdoc, Ben Cater, was a good friend of Roger. And actually, Roger, when I got my first faculty position at University of Kentucky in 1989, he came and visited my lab. Um, but anyway, so we were one of the first labs to be able to use his fluorescent calcium indicator probes in living neurons. And this was an experiment I did with another postdoc in the lab, Peter Guthrie. And so what we did in this experiment, I introduced this fluorescent calcium probe into cultured neurons. And this is just showing one neuron here in a culture. And then what I did is I took a micropipette and I cut the axon so that the distal region of the axon is physically separated from the rest of the axon and the rest of the cell. And sometimes when I make this cut here, this distal region and the cell will survive nicely and the calcium levels remain low. So in this image, blue represents low calcium levels, and then green, yellow, and red are increasing calcium levels. So we took an image of the calcium levels, uh, and uh, and then I added glutamate to the culture. And what you see is that calcium levels rise in the dendrites, the cell body, and then there's a decreasing gradient of calcium in the attached portion of the axon. But there's no change in calcium in the part of the axon physically removed from the rest of the neuron. And the reason is there's no receptors in the membrane of the axon for glutamate, whereas there are receptors in the dendrites. Um, and then we did kind of a control experiment to make sure that we can detect the calcium change in the axon. So we depolarized the membrane by adding high potassium outside the cell. and we got elevation in calcium. So that was a kind of a cool experiment at the foundations of understanding calcium's role in regulating neurons. Okay, so this study is essentially what they show using imaging methods, uh, where they're able to label actin and mitochondria in the same cells, and they're able to stimulate very precisely increase in calcium levels in cells. Um, what they show is that when you expose a synapse, a dendritic spine, to calcium influx through glutamate receptors, that calcium influx through this kinase that I mentioned, CAN kinase 2, um, leads to the stalling of the mitochondria there and keeps them at the synapse. Okay, so calcium influx stimulates BDNF production, and some of that calcium can probably actually come from the mitochondria. Um, and when the synapse is activated and you get calcium release from the mitochondria, and that's thought actually to contribute to signaling both at the synapse and by activating transcription factors like CREB that I mentioned that stimulate production of BDNF. In addition, of course, mitochondria produce oxygen free radicals or reactive oxygen species. So remember, the oxygen that's not used in the electron transport chain is converted to superoxide anion radical, and then it is converted by superoxide dismutase 2 to hydrogen peroxide, and hydrogen peroxide can be, in the presence of iron or copper, can be converted to another free radical called hydroxyl radical, which is very damaging. But there are enzymes that remove hydrogen peroxide and detoxify it with well. But there's now good evidence that superoxide and hydrogen peroxide have signaling functions. And actually, there's another free radical called nitric oxide, which is a gas and can diffuse readily. Um, Nitric oxide is produced in response to calcium influx. And there's a specific pathway involving an enzyme called nitric oxide synthase. And nitric oxide was discovered and got a Nobel Prize for the work because it's the key thing in causing relaxation of your blood vessels in response to certain stimuli. So it can lower blood pressure. In fact, with someone has coronary artery disease and they have chest pain, sometimes they're given nitroglycerin, right? Under the tongue, for example. And what happens there is the nitroglycerin, you generate the gas nitric oxide, which then acts on the cells surrounding your blood vessel to cause relaxation. Well, it turns out that nitric oxide, which is produced when neurons are active, um, in response to calcium again, there's evidence it plays important roles in synaptic plasticity and learning and memory. And Saul Snyder at Johns Hopkins, my mentor there, actually, um, he did a lot of work, the early work on nitric oxide in the nervous system. All right. Now, remember in the first presentation, I talked about the citric acid cycle, the TCA cycle, and how glucose is used to generate ATP, get TCA cycle, and then use electron uh, donors act in the mitochondrial complex and so on. And during this process, NADH is produced, the reduced form of nicotinamide adenine dinucleotide. Now, it turns out that the oxidized form of NAD, which is NAD plus, so loss of an electron, oxidation, you get a plus charge. NAD plus has signaling functions. There's a lot of work going on with NAD. It's involved energy metabolism, but it also has signaling functions. And I wanted to mention them before I conclude because next presentation, when I talk about exercise and fasting, I'll be talking about NAD plus. Um, yeah, I'm gonna skip this because I kind of get it. All right. So NAD plus uh, uh, plays a role in the activation of enzymes called sirtuins, which are protein deacetylases. They remove an acetyl group from amino acid lysine in proteins, and that affects the function of the protein. And these sirtuins play important roles in a lot of normal cellular functions. Their responses to environmental stimuli. There's evidence they play a role in aging, and we and others are finding they play very important roles in the brain. And these sirtuins, some of them, the levels of the protein itself or the activity of the protein are affected by both good and bad types of environmental exposure. So good exercise, intermittent fasting, caloric restriction, for example, intellectual challenges. And then the bad things would be chronic psychosocial stress, for example, or in aging or neurodegenerative disorders. Okay. Um, yeah, and so I'm going to finish up with this. This is a diagram showing the so-called sirtuin family. And there are many sirtuin proteins. So these are proteins with similar amino acid sequences, and many of them do similar things. For example, function as deacetylases. The first one that was really looked at a lot was SIRT1 or sirtuin 1. And it was actually discovered in yeast, where it was found to play an important role in preventing or slowing down aging, if you will, in the yeast. And there's quite a bit of work in mammals, mice and cultured cells, showing its important roles in responses to metabolic challenges, other types of stress. So it seems to be important in stress. It affects gene expression. It's present in the nucleus, some apparently also in the cytoplasm. One we've studied a lot, which I'll talk about next time, is sirtuin 3, SIRT3, which is located exclusively in the mitochondria. And it affects a lot of things in the mitochondria. It affects ATP production, uh, levels of free radicals. It affects also other processes, calcium regulation, for example, and it can even protect cells from dying by its actions in the mitochondria. And this final slide here actually is just an example of some of the proteins. There's been several hundred proteins that is thought to be regulated in the mitochondria by SIRT3. Some are involved in energy production, some are involved in amino acid metabolism, um, some are involved in ketone production. Remember, the ketones are produced from fatty acids uh, in the mitochondria, and you know, reactive oxygen species, and so on. Okay, so I'll finish actually with this slide, and I'll probably start with it next time. And this has to do with metabolic switching from glucose to ketones, how that occurs, uh, and essentially involves with extended exercise or fasting, you get uh, release of fatty acids from the fat cells, your adipocytes. These free fatty acids then go into the liver cells and converted to ketones, beta hydroxybutyrate, acetoacetate. These are actively moved into cells, including neurons in the brain, where they can be used to produce ATP. Remember, this fatty acids are broken down to acetyl-CoA, which is then incorporated into the ketones, which are then broken down to acetyl-CoA, which goes into the TCA cycle, generate ATP. And turns out, though, very interesting, very recent findings, this ketone, BHB, affects gene expression uh, through and protein function through several ways that are being discovered. We discovered, for example, that can increase levels of BDNF cells. Okay, so that's all for today. I hope you followed most of this and learned some of it. And by the way, you're welcome to contact me either through the comment section in YouTube, or you can email me if you want to at my Johns Hopkins email address, which you can find. So thanks. I hope you have a good time till the next time you visit Brain Ponderings. Goodbye.